Okay, good evening. This is Dr. Ken Shen from West Virginia University. Today will all be our last section, talk about biochemistry genetics, and we will talk about RNA translation. So let's still review the central dogma. And remember I said, Crick mentioned this in 1958. It is always a flowchart for all the genetic knowledge. Let's do it in a different way. So I have a DNA sequence, 5' to 3' and 3' to 5'. This is running opposite direction. Let's say this is DNA. And going through the replication. And let's put some of the sequence there. So I have ATG, GGG, and TTT, let's say. Now, what's going to be our corresponding complementary band the sequence looks like? It will be T, A, C, 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 and A, A, A. Based on gene complementary policy, and these are all connected with hydrogen bonds. And we all know these things already. Now, we're gonna going through is RNA transcription. So, we will get a sequence which is mRNA. What gonna be looks like? It will be complementary to one of the bands of DNA is 5' to 3'. So remember, we have two bands here. The top one we call it a coding band, and the bottom one is a template band. And this RNA sequencing should be exactly the same as a coding band. The only difference, it will be instead of you have T, thymine, you have a uracil, which is U. So this becomes AUG, GGG, and UUU. Okay, we're gonna move to the last section. We call it translation. Now, what's gonna be looks like? We have the genetic code there. So at the end of the day, they will become amino acids. What are they? We had a genetic code. It's right here. It's on the table. Each 3 letter representing amino acids. So what is AUG for? Is MET. Methylcyanic. What is GGG? GLY, glycine. What is UUU? PHE, phenylalanine, and they are connected. And which one is carrying these amino acids, if you still remember? It is we call tRNA, transfer RNA. Okay, so each of them will be carried on by a tRNA. And 5', 3', and goes here. We can draw all of them, and we'll talk about this later. 5' and 3'. Okay, these, do not forget, all these tRNA is carry on anti-codon. It is not a codon, it's an anti-codon. Well, what are they? This is going to be UAC, the second one will be CCC, and the last one will be AAA. So, this is called the translation. Now, today, we're going to talk about detail of the translation. Before we talk about the translation, we would love to go over 2 basic knowledge. Number 1 is amino acid. because at the end of the day, they'll become amino acids. So we need to know what is amino acid. So said very simple. Amino acid is a structure. Is always like this. In the middle is a C connected with a proton, which is hydrogen. And then you have a left side, NH2. This is amino group. Right side, you have COOH. This we could also write like this. So we call it carboxyl group. Now, how are you understanding about that? This is a structure. We call it carbo. OH is hydroxy base or bonds. The hydroxy we don't mention it. So we connected, call it carboxy group. Then you have an R. What is this one? This R is a side chain. And this will be decide what type of the amino acid it is. So I give you some of the example. If this R is H, it's the simplest amino acid is glycine. If this R is CH3, which is a methyl group, this will become alanine. Now let's do something a little bit complicated. If this R is a CH2 combined with a phenyl ring, this is we call phenylalanine. Now, let's say this R is CH3 with a sulfide S. Then we CH2 and CH2. This is called methylcyanine. Okay? Sorry. methionine. This is methylcyanine. Now, all of these, we have an acronym, GLY, ALA, PHE, and MET. Okay, when you look at the structure, this is called amino acids. You should know this side, the amino group is positively charged. And the right side, the carboxy group is negatively charged. Amino group right there, obviously is amine group. Do you feeling about here, the carboxy group kind of like acid? It's not really acid, like acid. So we want to talk a little bit more. All of these two ends, it's a biochemical reaction, which means there's some story going to happen there. Why? How they connected with each other. This is covalent bond. Do you remember when we talk about water, this is also covalent bond. But you know, water is much more stable. The only way to break down is you going through electricity. But amino acid base is easily to react sometimes. Why? Because the electronegativity of all oxygen is 3.5. And the electronegativity of nitrogen is 2.8 to 3.0. it is a little bit lower. Therefore, compared to the water molecule, this amino group is a little bit more attracted by a biochemical reaction. But you know amino acid is still very stable most of the time. So you need to know a little bit behind this. Now, who mentioned about this electronegativity? A great American scientist and chemist, Pauling. And now when I talk about DNA replication, I said he also finds kind of the twisted helix, almost getting there. But at the end of the day, it's Crick and Watson cross the finish line. Okay, so this is something I want to mention, the amino acid. The second thing, we talk about this tRNA. So we want to mention a little bit about this tRNA. What it looks like, transfer RNA. You know that, we're going to talk a little bit brief. This is always like a tree leafy. Okay, this is what it looks like. And we have 3' ends and 5' ends. There are so many arms there. This is the first one you know, it's anti-codon arm. Carry-on anti-codon. This side, I said, they always have some very weird, strange gene base. Let's say this is T theta C arm. This side, we call it D loop, or we say D arm. How about this side? Usually, we will be connected to amino acid. We're going to talk about detail later on. This is called amino acid accept arm. Now, sometimes, some of the textbook also will say, there is a 3 tiny arm here. This is we call variable arm. But regarding the RNA translation, this anti-codon arm will be the key. And this amino accept arm will also be the key. These we're going to mention later on. Okay, so these are 2 basic knowledge we need to know before we talk about detail. Then, let's talk about the genetic code. Okay, we want to talk about that. So the third section we want to talk about is genetic code. So first of all, what is genetic code? Genetic code, which means the 3-letter code, each will be translated become an amino acid. So I use AA representing amino acid. That's called a translation. And I said what is called a translation? What I implicated? Letter to amino acid. If letter to letter is called a transcription. Now, before we talk about detail, let's do a simple math. Okay, I had 3 boxes. Box number 1, box number 2, and box number 3. I labeled them 1, 2, 3. Every box, there is a possible, could have 4 different types of the gene base, which is A, U, C, G. So adenine, uracil, thymine, and guanine. Now the first question, how many possible genetic code will be created theoretically? Remember what I said is theoretically. You can do a math. And I told all my students, you get accepted by West Virginia University, you should be good at this math. And you all from China, and you're at Nanjing Agricultural University, you're doing the math puzzle since you're 5 years old. You should be better than them for the math. So let's do it together. What's the possibility of the first box? It's 4. What's the possibility of the second box? Is 4. How about the third box? Is also 4. Because each of the box could be A, U, C, G, one of them, and they could be repeated that. So what's possible theoretical number? You multiply them together. 4 multiplied by 4 multiplied by 4, which means 4 times 3, that is 64. Okay, there is 64 theoretical genetic code. Now we have a problem. You will say, Dr. Shen, what I learned in my biology in high school, or biology 101 in my undergraduate class, we said it is 20 amino acid and there is a 61 genetic code. How come you come up with 64? So let's talk about this. 64 minus by 61 equals 3. What are these 3 extra? These 3 extra are very important. What are they? That are UAG, UGA, and UAA. These we call stop code or stop code. Because these are the signal to tell you they're going to be stopped, the translation. So those are the code are important. Okay, now we have another question. You said at the beginning, 3-letter code translated for amino acids. I had 20 amino acids. So I should have 20 genetic code. How come instead of have 20, I have 61? What this means? That means one amino acid could be translated from more than one, I will say 1 to 4 genetic code. Now why? Because we have these 3 boxes. We box them, we label them, number 1, number 2, and number 3. We are more strict for the position of number 1 and number 2. We give them a little bit of flexibility of number 3. And this phenomenon we call it wobble. This is very important. Why? We mentioned about that. During the RNA replication, this DNA polymerase is very efficiency. It can do 2000 BP replication per second. They will have some mistake, which means there is some mutation going to come out. Now we had proofreading by our DNA polymerase 3. We have a mismatch repair system. We had an SOS system, but we're still going to have a mistake. And the very important, the body of the bacteria gave you some flexible. That's called a wobble. Which means if you have some of the mutation happens or mistake or errors happened for the third box, it's not going to affect you the final genetic sequencing and what type of the amino acid comes out. This is called a wobble, and it is very important. So this type of mutation, we also call it silent mutation, or we call it nonsense mutation. So let's give you an example. We said GGG is glycine. Is that right? What I said GGG is glycine. If you look at the genetic code right here, you will see GGA, GGU, and GGC, these are going to be translated to glycine. So that's an example of the Wobble phenomenon. Okay, let's back to here. We have 20 amino acids, 60 genetic code corresponding to that. That is called what? Redundancy. If we use a layman language, we have a scientific terminology for that called degeneracy. Okay? A scientific name for that. Last thing I want to mention, around here. Genetic code is very universal, which means from virus to bacteria to animal cells to plant cells, this set of the genetic code right here fits all of them. There is only one exception for animal, is the mitochondria. If we talk about genetic code inside of mitochondria, it is different. Okay, so just I want to tell you one thing. Most of them are universal. So this is we talk about genetic code. Okay, we finished talking about these basic knowledge. We're gonna go ahead to talk about detail of the translation. When we talk about translation, very similar to before. Also 3 stages. What are they? Initiation, elongation, and termination. Now, all of these in the translation process, they all need some factors, which means they are proteins. Initiation need IF factor 1, 2, 3. Elongation, they need elongation factor TU and G. Termination, they need a releasing factor 1, 2, 3. Those are all proteins. So we will talk about detail of all these. Okay, the first thing, number 1, initiation. Okay, before we talk about initiation, we need to go back to talk about the structure of tRNA, and we need to go back to talk about the amino acid, and then we also going to talk about the RNA and what it looks like. Okay, we're going to go back talk about some of the structure. So first of all, we mentioned this tRNA going to carry on amino acid. How they connected together? Okay, how these things going to be work? So we're going to go back to look at the RNA. Let's draw the structure of RNA. What it looks like. 5 carbon ribose. This side will be carry on a gene base. Let's say it's A. Okay? And then we have H, OH, H, OH, H, CH2. And I said go up is always going to be important and going to be beautiful, because we will be talking about is a beautiful phosphates. This is important. Okay. First of all, we're going to be the tRNA goes here. This always going here is a tRNA. Start from the O side. Okay, how are we going to draw it? We will draw right here. This is a tRNA. 5 prime and 3 prime. How they connected? They need something help. Some of the textbook will using a C to marker them. They need 2 thymine as the initiator to connect it an RNA, which is an RNA nucleotides. Where going to be the amino acid going to be coming? Of course, this is the anticodon. We're going to go back to top. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. The story going to happen is in the 3 prime. Okay, how they do? Remember, this is the basic structure of an amino acid. What is it going to be looks like? Okay, R, C, OH, then goes, I will just draw like this. C, R, NH2, H, C, O, O, H. The story gonna happen is right here to this oxygen of the 3 prime ends. If they add together, we'll have a water come out. And then this is gone. The hydroxy base is gone, and this H is gone. So how they connected? They actually connected with this O is a high energy bonds. This is a high energy bonds. What their storage? Kind of like a phosphates. This process, we need ATP hydrolyze, become AMP, and 2 phosphates. This is also need the help of magnesium. What is this whole process called? Amino acyl synthesis. What is the enzyme responsible for this? The enzyme responsible for this connection is very simple. Is amino acyl synthesis. When you see ASE, it's an enzyme. Okay, this is the first thing you need to know. Is what is called amino acyl synthesis. This is number 1. Some basic knowledge. Okay. Then we talk about initiation. Okay. First question. Where are the place happened the translation? And said very simple, where the place happened is the protein synthesis. Always remember is ribosome. And I want to also mention ribosome is a very few cell machinery, both existing in prokaryotes and eukaryotes microorganisms. We still using E. coli as an example. E. coli is 70S ribosome. They have 2 units. What are they? 50S large units and 30S small units. And I said they are not add up together because the S is not molecular weight. It's a Vandenberg precipitation speed, we call it velocity. That's why they're not add up. Underneath 50S subunits, we have 5S and 23S rRNA, ribosome RNA. And underneath the 30S small subunits, we have a 60S rRNA. So we very briefly go over these. Okay, then we talk about the initiation. What are going to happen? Let's draw the picture, talk about that. First of all, there is a 30 subunits there. This is 30 subunits. At the beginning, you need to know, before they start translation, these two are separated. They are not connected together. How are they separated? We mentioned about the initial factor. Initial factor 3, IF3, is connected to the ends of the 30 subunits to make sure they are separated. Now, once we started the translation, this guy gone. Okay, they're gonna be isolated. So this is IF3. Stay on the side, stand by. Now, what else gonna be there? Initial factor 1 will attach to the 30 subunits. Okay, then we start to do something. Then what happened? We had a mRNA gonna come in. Now, first of all, you need to know. Oh, this is rounding 5 to 3 pi. Does not mean any mRNA will going through translation. They have some signal. What are the two major signals? Number one, everybody use, you need to have AUG. Because AUG is an initial code. So you have an AUG. This is everybody knows. The second thing, do not forget. In the upstream of the AUG, they need to have some very special sequencing, which is AGG. This will be recognized by the 60S rRNA underneath, inside 30S subunits. Now, what is AGG called? This is called Shen NAU sequence, which is called SD sequence. So said very simple, this is SD sequence, which has to be recognized by 16S rRNA. Okay, once you have these things ready, there is an tRNA going to carry on something coming. Okay, what are they? I'm going to draw a little bit simple. This is the tRNA, will be carried on by the first one. Usually it's MET. AUG translated to the MET, methylcyanine. This guy usually carry on by an initial factor 2. And this initial factor 2 at the same time bring the energy money, the energy currency. It is GTP. Very simple understanding. Initial factor 2 is like a car we're going to drive in. I'm carrying on there, go to national park. I wanted to join the club, is that right? I need to pay the membership fee. At this moment for bacteria is GTP. So what happened here? At the end of the day, this will become GDP and PI. Okay, then this guy gonna come in. So what's gonna happen later on? This is hydrolyzed, we don't need that. Okay? This guy will be moving to here. Right in the center. We'll talk a little bit detail later. Okay, they're moving into the center, and then a large 50S subunits will be put it on. At the same time, this guy is gone. The initial factor 3 going to be released. So let them to start. Okay, I'm going to talk about something in detail. Not that easy. First question. Every time they are carrying the first genetic code is AUG, translated to become amino acid is MET, the methylcyanine. But I want to tell you there's some difference. If it is a bacteria, the first one we call tRNA is carrying FMET. If it's a fungi, which means the eukaryotes, this tRNA, the first one is carrying is MET. No F. So what's that? Right here. The difference is this R. What's the difference of this R? I'll just do some connected to here. Okay, a little bit of connector here. What is FMET looks like? This is called formyl methylcyanine. What's that? CH3, S, CH2, CH2, go here, CH, this COO, and tRNA, FMET. So what is that one? Go down, NH, amino base, amino group, go here, CH, CO, and CH. This structure is formyl. If you don't know this, do you still remember this guy? What it looks like? Aldehyde, is that right? Very similar. Now, this is FMET is for bacteria. For fungi, we don't have this one, so we remove it. Okay? This is MET, methylcyanine. And also, of course, for fungi, we don't have that F. So we remove that. So this is the first thing you know. The second thing we also want to mention, they go right in the middle. Because inside of this ribosome, there is a 3 location. This 3 location called EPA. What are the EPA? This is not Environmental Protection Agency in the Washington, D.C. These days we talk about climate change or environmental protection, sustainability. We talk this agency all the time. Some people said we want to get rid of that. It is not an Environmental Protection Agency. What is that? Exit site. P, peptidal site. A, amino acid accept site. Some of the textbook says amino acyl site. Same thing. Now, in the P, which is peptidal site, this is happened is transpeptidation. And this need an enzyme called transpeptidase. This is coming from a 23S RNA inside 50 subunits. So this is from 23S RNA. And this is one of the units, one of the components is from 50S large subunits. Okay, so you need to know what the EPA looks like. Then we're going to talk about. So this is initiation. First thing, big A. Okay, second thing, we want to talk about here. Let's draw on the top, because I'm going to using probably the whole. Elongation. T-I-O-N. I'll take this route because you already know about it. I draw the line here to make you know more clearly. Okay, so B elongation. What's going to happen? Okay, we already initiated. So this is 30S. And we already have this guy there. At the P location, this is E and this is A. Okay, I already have an amino acid, AA, and the initial one always is MET. Okay, now of course, this is packaged by 50S large subunits. Then we start to have this long chain to synthesize. First of all, something gonna come into the A. Let's say we have an AA, amino acid. This is gonna go there. Okay, something gonna come. Now what happened? The second step. We're gonna draw here. 38 subunits. 3 location, EPA. Okay. What's gonna be? This will go here. Okay, so what it looks like? This P will be empty. The tRNA will be empty. On right side here, you will have AA, and MET on top. What is this one called? Transpeptidation. They require elongation factor PU. Also need GTP, hydrolyze become GDP, and phosphates. Okay, now of course we have this guy. This 50S subunits. Okay, next stage, what's gonna happen? Go next one. I'll have right here. Okay, still EPA, 3 location. This stuff is gonna move. Goes from right to left direction. 5' to 3'. Okay, goes there. What's gonna happen? This guy, the empty tRNA will go to the E. This gonna move in. This guy gonna move to here. So you connect it with AA, and with MET. And of course, there is a new one gonna come in. tRNA carry on AA. Well, now of course you had a big package, 50 subunits. This is serious. Okay, next what's gonna happen? Next one, if we draw the last one here, what's gonna happen? We still have EPA, 3 location. Okay, first of all, here the empty tRNA will release. Because when they at E, we call it exit site. Once we say exit site, let's find what I write there. Okay, I already removed the exit site, which means it's gonna be gone. So this is empty tRNA will release. They are not just to go there to have fun. They will be looking for another amino acid and later on gonna go here. Okay? What are the others gonna do? This gonna be move to here. Is that right? Then what happened? This guy will be moved to here, then become AA, sorry, not this AA, the purple AA, then AA, then we have this color AA, and the last one is MBT. Then a new one gonna come in. Carry on another color. Okay, this process, we call it translocation. This translocation need EF, G, elongation factor G, also need GTP, hydrolyze become GDP and PI. Now, you're a little bit confusing about what happened here. I know about what happened here. So, we're gonna explain to you right here, what's this gonna be? Okay, this is P site. It's gonna be A site. Let's say on the P site, we already have a 5 carbon there, okay? This is, let's say a the H, OH, then goes here. We're gonna have O, goes here, we're gonna have C, O, then we're gonna go here, we're gonna have R, N, plus 1, and then go H, and NH, which is amino base, so go down, we have C, O, and then go here, C, H, and then we go, sorry, we have another O right here. Yeah, that's correct. Then R, N, and then go here, have NH. Very long. Now this side, CH2, go, O, phosphate, O, then connected with a tRNA right here. We draw the picture. I said never hesitate to draw. We have C, this is